So, I'd like to welcome you all to the symposium on dietary biomarkers for measuring food habits, possibilities, and challenges. This is a two-day symposium, and we are very happy that we have been able to attract such interesting and highly qualified speakers for the symposium. So I think we can all look forward to two very interesting half days. My name is Anna Winqvist. I'm a professor in nutrition from University of Gothenburg. The symposium is organized and hosted jointly by 3 different organizations, and I represent 2 of these. I'd like to introduce them. So first we have the Royal Swedish Academy of Sciences, and the Academy has 18 national committees, and the Swedish National Committee on Nutrition and Food Sciences is one of them. And these national committees, they represent Sweden in international organizations. In our case, we represent Sweden in IUNS, the International Union for Nutrition Sciences. And we also are a link between science and society, and we often give advice to universities and to the Royal Academy. We consist of 18 members, professors at different universities and institutes in Sweden that represent nutrition and food sciences, and we annually arrange international symposia. So we are very happy to be part of this symposium here on dietary biomarkers. We also have the Swedish Network in Epidemiology and Nutrition. This is a national network that has existed since 1985. It's funded by the Swedish Research Council for Health, Working Life and Welfare, and we currently are about 450 members. The network is open to anyone working in Sweden and with an interest in nutrition research and methodological issues. And also this organization annually organizes national and sometimes international meetings on topics related to nutrition research. We have a working group consisting of 13 members, again representing most of the universities and institutes in Sweden in this research area. And it's members of this working group who arrange national meetings. And this year it has been Ulrika Eriksson and Emily Sonestedt from Lund University who have been the organizers on behalf of NEON. So with that, Ulrika, I leave to you to introduce the third party and also the program. Thank you.
Thank you, Anna. So my name is Ulrika Eriksson, and I'm also very happy to welcome you to this meeting called Dietary Biomarkers for Measuring Food Habits, Possibilities and Challenges. And I hope that we will learn a lot today about dietary biomarkers and also about their potential use in future nutritional epidemiology. And as Anna said, I've been organizing the meeting together with Emily Sonestedt and the Royal Swedish Academy of Sciences and the NEON Working Group, and also together with other researchers at Lund University and EpiHealth. And EpiHealth is the strategic research area epidemiology for health at Lund University and Uppsala University. And their ambition is to strengthen collaborations between researchers in this field. And now to the program. And I think we have a very exciting program today. And we have speakers from all over the world, from Denmark, Sweden, Germany, Ireland, the UK, and Australia. So please take the opportunity to ask questions today. And you can do that by clicking the Q&A button in the webinar and write down your questions. And then there will be time after each speaker for those questions. And there will be moderators asking the questions that you put in the Q&A. So please do that. And there will also be short breaks after every second presentation so that you can stretch your legs. And the meeting will be recorded, and it will also be running on the Royal Swedish Academy of Sciences YouTube channel. So please enjoy the meeting. And now I give the word to Emily Sonestedt.
Yes, hello. I'm also very glad to see so many participants. So before we start the meeting, we would like to know more about you that are listening. So we have a few questions to ask you. And the first question is, which country do you live in? Sweden, other Nordic country, other European country, or country outside Europe? So please press the answer. We can see 85% in Sweden, some in Nordic countries, and actually 2 participants outside Europe. It's really, really nice to see. Take the next question. So are you a NEON member? Yes, no, or no, but I want to become a member. So 54% are NEON members, and yeah, 24 participants that want to become a member. Nice to see. So the third question. So where do you work? Academia, healthcare, food, pharma industry, no employer, I'm a student, or other. So 69% in academia, and 6% as a student. Nice. Now we know more about you and each other. So now I give the word to Ulrika, who will moderate the next session.
Thank you, Emily. First out is Professor Lars-Uwe Dragstedt from Copenhagen University, and he will give an introduction to dietary biomarkers, as he has very broad experience in this field. And he is also one of the leaders of the multi-center project Foodball, where one of the aims is to discover new biomarkers or new dietary biomarkers. So please take the opportunity to ask questions during his speak. And yes, please start, Professor Dragstedt.
Thank you very much. Just a moment, I'll make it presentation mode. So here we go. So I was asked to give, first of all, I'd like to thank the organizers for inviting me. I'm very pleased to give this introduction, which means I'll give some of the new ideas, some of the new things that we have developed in the Foodball consortium that you just mentioned, which was European, but it also included members from Canada and a few other places, which try to work on dietary biomarkers and develop as many as possible. I'll come back to how we worked on that. As you know, the current state of assessing food intake has some limitations. We have subjective tools like dietary history, food frequency questionnaires, and weight dietary records and 24 recalls, which are all kind of time consuming. They are based on memory and composition tables, focused on specific foods and nutrients and on specific populations. So they may be difficult to employ. They also have systematic and random errors. And I think it's fair to say that we actually don't know to what extent. At least I've been surprised during my research to see that for some foods it was not as bad as I thought. For others it was really bad. So it probably depends a bit. Very, very simple basic model of biomarker classification is consists of these three modalities. So you have the exposure to whatever, you have the system susceptibility, you could also call it health or resilience, and then depending on how that modulates the exposure, you would have a certain effect. Very, very simple model that can be employed everywhere. I mean, even in environmental science. Now, when we look at humans, they are actually a bit more intelligent, say. So they may actually respond in a way so that they change things as a response. If you notice that you are taking on additional weight, you may actually change your habits a bit and thereby also your susceptibility. So we have a more complex system, of course, as soon as we deal with humans and society. This is another way of looking at it, just to show that we have many different kinds of exposures. So, of course, the dietary exposure is one, but the dietary exposure is actually multiple. I mean, you have the chemical exposure from the food as such. You have the physiological effects there, but you also have a lot of psychological effects around the meal and other things. So you have many different exposures as part of eating. So therefore, I mean, remember that you have a lot of different exposures all the time. These exposures will have a certain effect, but the effect that it will have on a given individual will depend on the health and susceptibility of this individual. So I've tried to illustrate that with these levels here that are pressed more or less depending on the system. Now, today, of course, we discuss mainly the exposure, but it's important to mention that it's not really the biomarker that, or the measurement as such, that determines whether you measure exposure, effect, or health. It is much more the intention of how you use it. So, for instance, if you measure glucose, that could, of course, measure the recent intake of glucose, but it could also be a response in general that if you measure it in the morning would be related to your health susceptibility. If you change your dietary habits and measure it again, you'd see the effect. So the same measurement, but different purposes. This is maybe one of the most important changes in the football definition of these different modalities in biomarker research, that it depends on how you use a biomarker. Now, when we look at the food intake biomarkers, then you can say, okay, what do we talk about? Is it nutrients or non-nutrients, or is it a whole food, a food component, or is it the dietary biomarker? You could also ask, I mean, what is the current status of a certain vitamin or something else? And then we are actually more in the health area, because that will depend on how much you've been eating for quite a while, and it will also be on the interaction, so to speak, two legs of this figure, where you both have exposure and health or susceptibility. So you can have your nutrient status down here, and you can have also non-nutrient status and of course other things that are more related to your individual risk. And the effect is then a third area here. But there is an overlap, so these are not absolute, and you can subdivide them in many different ways. So also, you have to remember that there is quite a complex food ontology. I mean, we all talk about diet, and the diet is, of course, complex, consists of a lot of different foods, and these foods can be subdivided into or assigned with different food groups or subgroups. So you have a lot of foods that are similar. So when we talk about developing biomarkers or finding biomarkers, a lot of these foods are quite similar, and we have to think about whether they overlap and what are we actually measuring. Are we measuring fruit, or are we measuring citrus intake, or whatever? So you also have meals, of course. The diet consists of meals, and each of these meals will have an impact for some time after the meal. And that, of course, will mean that the foods that the meal consists of will be somehow measurable in your body fluids like blood or urine. And this may depend on the ingredients and food components that exist in that food. So we have the bread, for instance, you have the dough, you have some salt, you have some different components like carbohydrate and lipid and protein, minerals, so on. But if you go beyond that, then you actually have the chemistry also. So you have the starch, you have the, if it's containing barley, for instance, you may have hordenine, have maltose. You could also have certain lipids. So you, the further you go down into it, the more detailed the chemistry picture becomes. So when you talk about biomarkers, you really have to take into consideration that there are very many levels of complexity here. So the food groups that we work with in football were these very broad major food groups. So you have a lot of them are plant-based, like the legumes, the fruit and vegetables, spices and herbs, and the non-alcoholic beverages. You also have fats and oils, many of which are vegetable origin, and you have cereals and whole grain. Then, of course, you have the confectionery and alcoholic beverages, which are more or less their own areas, but they can be quite complex and contain a lot of other things, so that they're not exactly the same. And then we put everything that may be too broad, the food of animal origin, which contains many different groups. We can subdivide it here and see, okay, we have the dairy, we have eggs, we have meat, and we have also subgroups within them. So red meat or fish and seafood. You have other terrestrial white meats like poultry, and you have offal meat. So when you talk about meat intake, you have to specify, and you also have to ask whether the biomarkers are actually covering all of these or some specific ones. So when you talk about food consumption, you can always ask this question, what do you mean? Is this the recent intake of one food, or is it average consumption of each food? In epidemiology, that's very often what you want. But it could also be a class signature of the diet, so kind of nutrityping or diet classification. So you have diet A, B, C, D, E, where you want to group your participants into these different groups. So you have to discuss very much. Do you want an individual assignment or a group assignment? Is it qualitative or quantitative? And are we actually likely to find biomarkers for all of these purposes? So I'll try to give you a little introduction to how far we got. And today, I think a lot of different groups in the world are working on this. So I'm pretty sure we'll get very far. So here are some compounds that were measured in a study of onion intake. These subjects got onion every day for 14 days, the same amount. But you could look at it after a single, the first single meal, or you can look at it after several days. And what you can see about these markers is that they have different half-lives. So they stay around, in this case, it's the excretion half-life in the urine. So they stay around for a different amount of time. And that is, of course, important. A very important part of the biomarker is how should you sample in order to measure a certain food. So the food consumption is, of course, very important here. Do you eat rarely? intermittently or frequently. So if you are a coffee drinker, you drink it every day, and you can live with a marker that has a short half-life. So I try to show here how you will have different sampling frequencies depending on the biomarkers excretion kinetics if you work with urine, or other kinetics if you work with blood or fecal samples. So, of course, the more frequently you work, the more rarely, or maybe only a single sampling, will be enough if you have markers that have a very long half-life. But this has to be taken into consideration when you design your and apply your biomarkers. You can also use several biomarkers for each food and thereby get more information about the timing, the frequency, and other things. This is a perspective that has not been fully developed yet, but it's definitely there to be explored. Then I'll go very briefly into metabolomics. I'm very certain the others will talk more about that, but it's been a fantastic tool to find and measure biomarkers. So it's, for those of you who are not familiar with it, it's just a way of profiling all the metabolites that are measurable by your instruments in a sample. So it could be, for instance, everything that has a molecular weight below 1500 Daltons in your biological specimen, and that flies in your equipment. It could be done by LC, liquid chromatography, time of flight mass spectrometry, or gas chromatography, or by nuclear magnetic resonance spectroscopy. And all of these have their strengths and weaknesses that I'll not go into. But the important thing to remember is that what you get includes both your endogenous and your exogenous metabolites. So a lot of things will change also as a response to the food, also your own endogenous metabolites. So you can go for everything that flies in an untargeted fashion, or you can go targeted for especially those things that you want to see. So the untargeted approaches are, of course, very useful for finding new biomarkers. But as soon as we know the biomarkers, we will go targeted, of course, to measure what we know we want to measure. This nice figure was made by Augustin Scalbert some years ago in a publication on the exposome. And of course, the food is only part of it. And it also affects other exposures. So that can be medical, or they could be pollution or other things. So you have the compounds that exist here, but they are digested. You also have the microbial metabolism, and you have the host metabolism. And then what you measure is then the food metabolome is then based on these changes, these changed metabolites that you find there. You also have your endogenous metabolome, as I mentioned, as the drug metabolome, pollutant metabolome, and so on and so forth. So you have very many metabolomes that go more or less into the same big pool when you look at untargeted measurements. So you can measure this in many different places, as I mentioned: serum, urine, stool, tissue, hair, whatever. Here's an example. This is made by another group some years ago, but it's nice for illustration. It's a marker of orange juice called proline betaine. Here's the structure. And as you can see, before the intake, level is low, and then it goes immediately up, and then it falls down again over the coming hours. So there are several things to note here. One is that the baseline level was not 0. So a lot of biomarkers actually have some background level. So in terms of measuring intake, trees do not necessarily grow into heaven. We will have some background level and some problems figuring that out. Another thing that you have to remember is that when people eat or drink juice or eat oranges, they also probably eat other fruit. Here I've, for 340 subjects, I've divided them into their general fruit intake and measured the proline betaine. As you can see, it looks like a very nice biomarker of fruit intake in general. So you are getting maybe more answers than you are asking sometimes. But of course, if someone eats fruit but do not eat any citrus, then we would not see them here, and there would not be a response at the individual level for that. So at the group level, it's very strong. At the individual level, you'll see something that varies a lot, goes up and down. Now, another example, this is related to beer intake. So we did a small meal study with different beers. So we have a control drink that was sports, I call it soft drink. You have light beer, you had a regular beer and a strong beer, and they have different levels of iso-alpha acids that come from the boiled hops, and they have different alcohol content. Some of them contain no alcohol, the others contain 5 and 8%. And we wanted to have a beer intake biomarker. That means we would like to see beer no matter if there was alcohol in it or not. So we just collected urine over a period of time after a single meet, also blood samples. It was a 4-way crossover, single-blinded intervention study with 18 healthy men and women, age 18 to 60 years old. So our hypothesis was that we could identify beer intake by markers, several markers combined. So we would like to have a dried barley germ biomarker, and that would be hordenine that I mentioned before. This compound exists only there. If you bake a bread of barley, it's not there. It's only from the germs. Also, the fermentation as such. And here, of course, alcohol doesn't work because we also want non-alcoholic beer to be. But we found out that ethyl malate seems to be a very good marker of this kind of fermentation. Then we have all the boiled hops compounds, the iso-alpha acids, but they are quickly oxidized in the human organism. So you get a biotransformation, you get all the breakdown products here, multiple products and different from person to person. So we'll have to have a whole profile of them alone. Using this, we could look into the population that we looked at here, and we could see that we had a very good marker. This is the area under the curve, but it was not perfect. And one of the reasons was that one subject here seemed to have high levels, even if in the control period. So we went back to this person, and this person actually started out with beer and strong beer, and then obviously knowing the structure of the study, could figure out that he would not have any alcohol the next 2 mornings. So he came in well loaded by himself, which was of course not the intention at all. We had tried to avoid anyone with alcohol problems, but we obviously got one here anyway. So we could see that at the individual level, we could easily pick up this marker, and we could see that this person had been non-compliant. Then we also looked into the a larger study and we tried to see, okay, can we discriminate between the different alcohol intake levels? So we have the strong beers here. This is just a PCA, so it's pretty easy to understand. It's just separation by multivariate statistics. You have this the wheat beer here, and you have the beverage without and the beer without alcohol here. So they separate completely. But if we look at the beer markers, then the wheat beer and the non-alcoholic beer, they are clustered together here. So we can separate the effect of alcohol from the effect of beer if we want to do that in further studies. So this is an example of combination biomarkers, which is very important in this area because we have to do that for a lot of different foods. Then the next I'll talk about is about validation. And you can see here a scheme that we developed in football, where we have 8 different characteristics. We have the plausibility. Is it likely this compound is a marker at all? The dose response kinetics, the time response kinetics, the robustness. Does this marker work when you eat all kinds of other foods? Reliability. Is this also in accordance with existing markers? The stability of the compound. Can we measure it after a long time in the freezer? analytical performance. That's a lot of different things actually, but that's what we are usually doing in the lab to make sure that our measurements are correct. And then the interlaboratory reproducibility. So this is what we call biomarker and validation. And we develop this based on what people say about validation in the literature. So all these things have been called validation, but combining them would actually be the way to fully validate a biomarker. So if we look at the markers existing, then here are some of the better validated ones. The allyl macapturic acid has been validated in several of these ways. We have proline betaine is pretty well validated. Today, one of the best actually. Ethyl sulfate from alcohol. You can see that chemical stability have for some reason not been described in the literature yet. So we'll have to go into that in order to finalize it. Then different fish markers like the DHA, EPA, and the special fatty acids that exist in fish that are called furan fatty acids that are degraded to a compound called CMPF. And you also have trimethylamine oxide, which exists in some, especially deep sea Atlantic fish. And you can see that some of them are not fully validated yet and need further study. There is also quite a lot of interest in guanidino acetate as a marker of chicken meat, probably also other birds. Although there may be some uncertainty whether this is in the feed to the birds or whether it's part of the birds themselves. It still needs to be exploited or investigated. And then something like curcumin, just to take one of the spices, you also have actually quite some markers for that. So I think there are a lot. These are not the only ones. There are a lot of other foods like whole grain rye and wheat, some legumes, oils, different meats, also smoked meats are pretty well validated at this time. So I'll just give you a few examples of some studies where we have had many subjects where we try to look into this. One of them for finding additional biomarkers, the other one for applying the biomarkers. So here's an intervention called UNINA, which is part of a dynamic consortium that we had, and this study was done in Italy by Danilo Acalini in Naples. And we had divided subjects into a control diet and a Mediterranean diet. So we looked at that. We had a baseline sample, and we had 4 and 8 weeks, and they were overweight and didn't have the best diet from the beginning. So here's just a single example here where we are interested in the elagitannins, which are compounds that are formed from a range of different foods actually. But in this particular study, we gave all the subjects walnuts. So here we see it as a function of walnut intake mainly. So we can see at baseline, there are some who are exposed to elagitannins, but as soon as we start the intervention, we can see that there is more being excreted. This is excreted with the fecal samples. If we look at the urine, we see more or less the same picture, although the variation was much higher at baseline, unfortunately, in the Mediterranean group. But still, there's no doubt that we see a major change as soon as we start with the intervention. So this also shows that sometimes you can look at other sample types and maybe get the relatively recent exposure from the urine, but you will have maybe a slightly longer term exposure from the fecal samples. Then last, since this is also a Nordic oriented meeting, we have Nordic diets. Here's the CIS diet study where Finland, Iceland, Sweden, and Denmark participated. And the inclusion criteria here were 30 to 65 years old and 27 to 38 in BMI, kilograms per square meter, and also one more metabolic syndrome parameter. And we had 96 subjects that were randomized to the healthy Nordic diet and 70 to the control diet. And there were samples taken at the baseline, 12 weeks, and either 18 or 24 weeks. And using metabolomics, we could easily separate people on the two diets. So we have a very good area under the curve here of 0.96. So only very few are not, and you can also see the separation here, although sin is less than the variables, it's partially cheating. But it actually was very efficient in separating the two groups. And here we have taken out all the markers related to the food items that we were interested in. So berry markers, whole grain markers, and others. But we didn't kind of select them. We looked at the difference between the two groups that we had before, and then we took those markers that were much higher in one group and much lower in the other group. And these markers are shown here. As you can see, we have the difference here between the control diet and the healthy Nordic diet. And we have a lot of markers that are related to the healthy diet, very few that are related to the control diet. Of course, if you eat something very white, starchy, fatty, and not very specific in terms of any fresh vegetables or fruits or other things, so an average Nordic diet, then there is not much to talk about, especially if you contrast with a diet which is rich in all the healthy food items. So we see a lot here and very little on this side. Still, you can actually give every subject a coordinate for their Nordic diet dishness, and then analyze your outcome parameters based on that. So this is a way of trying to look at whole diets, and we can see that there are a lot of markers here that are related to whole grain, some that are related to berry, and a lot that are related to fish. And depending on where you live in the Nordic countries, I mean, the Nordic countries span 2,000 times 2,000 kilometers, and therefore the foods are not the same all over. And we'll see differences in the fish, in the whole grains, and other things that were visible in the markers. So there is even more information in the biomarkers than we thought. So here the fish and whole grain direction. So qualitative validation of food intake biomarkers is important, and the biomarkers of food intake will be an important part of the dietary assessment. I don't think right now, and not even in the few years to come, that we are ready to substitute the subjective tools with biomarkers. However, we will be able to qualify them very much using these methodologies. I'll say there are still some critical gaps, and that is covering the different diets, the foods and their components with sufficient number of well-validated biomarkers, and also providing sampling strategies to allow efficient use of the biomarkers. So these are still things that need to be developed. So in conclusion, by accurately classifying, validating, and combining biomarkers, it is possible to detect the recent intake of several foods and drinks, identify misreport, and especially in short-term dietary assessment, detect dietary biomarkers in several sample types, and also look at biomarkers that are affected by the microbiome, and associate specific food intake biomarkers with clinical effects to form qualified hypotheses. And all this has been supported by a range of different funders. First of all, the Joint Program Initiative, where we had football and dynamic, also an EU FP7 grant, the Preview project, where I showed a few data from, Nordic Council of Ministers for the Swiss diet, Carlsberg Foundation for some of the basic development, and Novo Nordic recently on microbial biomarkers. So thank you for your attention. I had a lot of nice collaborators that are shown here.
So thank you so much for a very nice introduction. And I think we have some questions here. Let's see in the Q&A. How much urine do you need to do those analyses?
20 to 40 microliters. So not much.
Not much.
I think for epidemiologists, think about sampling many times. I mean, if you could get 10 morning urines, even a small spot, and make people put a small amount in their freezer, the pool of those urines would tell much, much more about the total diet than any single.
Yeah.
We still need to show that this is true in our scientific study. But I mean, from just basic logic, that would seem to be a much better way of doing it.
Okay, thank you. One more question. Thank you, Lars, for excellent talk. Have you compared alcohol biomarkers against the PETH? That appears to be very promising, but yet not explored for epi studies.
PETH we did not make, but fatty acid ethyl esters is something we're currently working on developing hair analysis for many of these markers, including alcohol biomarkers. And the ethyl sulfate and ethyl glucuride seem to have pretty good concordance with the fatty acid ethyl esters in hair, because then you have a more long-term assessment of the marker. It's a long story, but I mean, if you take the hair, then it grows slowly, and you have an average of over time of what you do.
Okay, thank you. And well, one more question came now. Thank you for an interesting presentation. You mentioned TMAO and of fish intake, but would it not be a biomarker of meat and other animal products too?
Well, depends on the population. If you have people who eat Atlantic fish like cod or herring mackerel, then you will see a lot of TMAO coming from that, because it exists directly in the fish. They use it as an osmolyte. If you have, depending on your microbiota, then you may have a higher or lower production of TMAO or trimethylamine from phospholipids and from creatinine, from other things that exist in meats. And then that will give you a more constant production of TMAO as kind of a background production that will be individual and probably is a individual risk marker. So you have it partly as an exposure marker for certain kinds of fish, but also as a probably increasing your risk of cardiovascular disease in terms of the formation in your gut.
Thank you so much. I think this was a very good introduction for the coming speakers. And I think that we need to go on to the next presentations. We have more questions in the Q&A, but we need to take them tomorrow, or if you could answer in the chat. So thank you so much. And now I would like to introduce Fredrik Rosqvist, and he will talk about biomarkers of fat intake. And since he is an expert on that, I think it will be a very interesting talk too. So please, Fredrik.
Thank you very much for the introduction, and thank you for the invitation to present. So I will be talking about using fatty acids as biomarkers of dietary intake. And fatty acid composition may be analyzed in various different tissues, for example, skeletal muscle or adipose tissue, but most commonly it is analyzed in blood samples, either in whole blood or in specific lipid fractions within blood. And the fatty acid composition is, of course, influenced by the type of dietary fat that we eat. But dietary intake is not the sole determinant. We also have endogenous metabolism. For example, we may synthesize saturated and monounsaturated fatty acids. And we may also desaturate and elongate fatty acids, meaning that a fatty acid that we consume may not necessarily stay as the same fatty acids inside the body. And this machinery of endogenous metabolism is in turn influenced by multiple different factors. For example, other nutrients. We know, for example, that carbohydrates, both quantity and quality, may affect the endogenous synthesis of saturated and monounsaturated fatty acids. Perhaps especially so if combined with a positive energy balance. And we also know that the genetic variants responsible for desaturation and elongation may influence the fatty acid composition, as well as the metabolic state of the individual. For example, if you have insulin resistance or type 2 diabetes. Thus, if you have data on fatty acid composition, if possible, this should be evaluated in light of the nutritional and metabolic context, perhaps in combination with self-reported dietary intake. It's important to stress that when using fatty acids, we look at the composition, not the amount. So we have no biomarker of total fat intake, but we do have good and useful biomarkers of dietary fat composition. Another important thing to remember is that fatty acids are typically reported as proportions, that is, relative amounts. So if one fatty acid increases, one or several others needs to decrease. Many fatty acids have been used as biomarkers, both for intake and for metabolic processes. This is a group, I've labeled them intake here. And here we can find, for example, the essential polyunsaturated fatty acids, which reflect vegetable fats and oils, and also nuts and seeds. And here we can also find the very long chain omega-3 PUFAs, EPA and DHA, reflecting intake of fatty fish. And we can also find here the odd chain fatty acids, such as 15-naught and 17-naught, commonly used to reflect intake of dairy fat. However, there have been quite some discussions about these during recent years, because now we have evidence that even these odd chain fatty acids may be endogenously synthesized using fiber, dietary fiber as a substrate. And observational studies have also shown that the proportions of these odd chain fatty acids are not only associated with self-reported intake of dairy fat, but also with the dietary sources of fiber. So these fatty acids are not as clear-cut as once believed. Another group of fatty acids, I've named them metabolic here, and they typically reflect metabolic events and not dietary intake as such. However, they may definitely be influenced by the intake of different types of dietary fat. And these are quite useful when looking at disease outcomes. However, we shall leave them for now. And then we have this group consisting of the common saturated fatty acids as well as the primary monounsaturated fatty acid, oleic acid. These fatty acids may reflect both dietary intake and endogenous metabolism. It is thus very difficult to disentangle their source of origin, and they are typically not very strong biomarkers of intake. However, some of these fatty acids may be useful as biomarkers in certain contexts and in certain populations. For example, we know from the EPIC study that the proportion of oleic acid was associated with self-reported intake of olive oil, but only in populations where the intake of olive oil was high. So this highlights the usefulness of evaluating fatty acid composition data in light of the overall nutritional context. And then we also have a fourth group of fatty acids here. And here we can, for example, find these very long chain saturated fatty acids having 20 carbons or more. These are typically strongly inversely associated with the disease outcomes. However, we know very little about these if they reflect dietary intake or endogenous metabolism or both. We will return to these in the end. As I mentioned in the beginning, fatty acid composition begins in whole blood, but perhaps more commonly is to use specific lipid fractions within blood. For example, phospholipids, cholesterol esters, and the red blood cells. The fraction of choice depends both on the context and the research question. You should be aware that the fatty acid composition differs across all these lipid fractions. This is just one example showing the composition in cholesterol esters to the left, which you see is clearly dominated by linoleic acid, and phospholipids to the right, where the major fatty acid is instead palmitate. Furthermore, all fatty acids may not be readily detectable in all lipid fractions. So if you have a particular interest for some minor fatty acids, that may guide you to which lipid fraction to use. So the dietary PUFAs are good and useful biomarkers. They've been used in many studies. And typically, we see a dose-response relationship with intake, and we can see this across lipid fractions. This is one example where each individual dot in this graph represents one study, and the x-axis shows the change in dietary intake of linoleic acid, and the y-axis shows the change in linoleic acid proportion in cholesterol esters. And this relationship is observed also in other lipid fractions, such as the phospholipids, although the magnitude of change likely differs between these fractions due to inherent in the lipid fractions. And there are many other examples. Here, example is a fish oil supplementation study where they provided 3 grams, 6 grams, or 9 grams of fish oil per day. And you can see that there is a nice dose-response relationship in the cholesterol esters on the top, but also in the red blood cells at the bottom. This shows the change in EPA. We also know that there is a strong correlation between these different lipid fractions. And this data is based on 8 Swedish studies, and the numbers that you see here are the correlation coefficients between the respective fatty acids in the phospholipid pool and in the cholesterol ester pool. And for the majority of fatty acids, you can see there are quite strong correlation coefficients between 0.7 and 0.9. For example, we have 0.8 for linoleic acid and 0.9 for EPA and DHA. However, for some fatty acids, for example, palmitate, there is a much weaker correlation. However, the take-home message from this study is that the proportion of fatty acids in phospholipids and cholesterol esters rank individuals similarly. So if you're ranked high in one fraction, you're likely to be ranked high in other fractions as well. And this is true for most fatty acids, although caution is advised when using some common saturated fatty acids. Okay, so we know that some fatty acids are good biomarkers, but what time frame of dietary intake is reflected? I think this is a very important question, and there's also some misconceptions about this, I think. There's a commonly held belief that some circulating lipid fractions are better long-term biomarkers of intake compared to other circulating lipid fractions. And the prime example here is, of course, the red blood cells having a lifespan of about 120 days. And thus it is commonly assumed that they will also reflect dietary intake during this time frame. This may or may not be true, and that will depend on the eventual changes in the dietary intake. we must remember that in most studies, we make the assumption that the subject has not made any drastic short-term changes. And I will show you why this assumption is so important. And the message I will try to convey now across the coming slides is that dietary intake rapidly influenced the composition in multiple circulating lipid fractions, including phospholipids and the red blood cells. This is a study where participants consumed a diet rich in saturated fat for 19 days, and then switched over to consume a diet rich in polyunsaturated fat, also for 19 days. Fatty acid composition was analyzed at the end of the saturated diet, and then after 1, 2, 5, 8, 12, and 19 days after crossing over to the PUFA-rich diet without any washout in between. And the authors could show here that when crossing over to the PUFA-rich diet, we see a rapid and marked increase in the proportion of linoleic acid in cholesterol esters, for example, to the left here, but also in phospholipids and in red blood cells. So for these, the phospholipids and the red cells, they could see a 12% relative increase already after 1 or 2 days after switching to the next diet. And the authors state that in the context of monitoring fat intake, the red cells provides no clear advantage over other lipid fractions, and that all these fractions show a similar rate of change as a function of time. Thus, neither fraction appears to reflect fat intake longer than the previous 1 to 2 weeks. And this has also been demonstrated when using stable isotopes. This is from a study where the participants were provided with a standardized breakfast meal, where the fatty acids in this meal were labeled with carbon-13. And fatty acid composition was analyzed in the fasting state, and then after 7 hours and 24 hours consumption of this meal. And we can see here in the cholesterol esters that after 24 hours, 5% of the linoleic acid in cholesterol ester was derived from this single breakfast meal. And in phospholipids, this was 10%, and this was evident already after 7 hours. So after 7 hours, you have turned over 10% of the linoleic in the phospholipid fraction, and this is then maintained up till 24 hours. And and also other fatty acids such as palmitate was also rapidly incorporated in the phospholipid fraction. And this was true also for the red blood cells, where they could see a rapid incorporation of both linoleic acid and palmitate after only 1 meal. The same group has also demonstrated that the maximal change is achieved already within 2 weeks. This is data from a crossover study where subjects consumed a diet rich in saturated fat and low in PUFAs, and then crossed over to a diet that was low in saturated fat and high in PUFAs, 8 weeks on each diet without any washout between. And when crossing over to the PUFA-rich diet, the authors demonstrate that the proportion of linoleic acid here in cholesterol esters to the left rapidly and markedly increased already after 2 weeks. And this was the same in phospholipids and also in erythrocytes, in the red blood cells. And you can see that the maximal change is achieved already within 2 weeks, and then these proportions are maintained throughout the remainder of the study. And when subjects crossed over to the diet rich in saturated fat, which primarily consisted of dairy fat, we could see a rapid increase in the odd-chain fatty acid 15-0, and this was true in cholesterol esters, in phospholipids, and also in red cells. And again, the maximal change is achieved within 2 weeks. Even adipose tissue may be influenced in a short time span if the dietary change is large enough. This is data based on 5 women who all changed their dietary fat composition to a high PUFA intake. And the authors here could demonstrate that the levels of linoleate in adipose tissue was significantly increased already after 14 days on this diet. and this was at the expense of oleic acid, which is the primary fatty acid in adipose tissue. And we have also demonstrated this in our own studies. This is based on 18 healthy individuals, increasing their intake of PUFA from 5 to 13 energy percentage as part of a 7-week overfeeding study. And here we can see a 19% increase in linoleic acid in adipose tissue across these 7 weeks, and significant decreases in oleic acid and palmitic acid. However, it should be stressed here that this was deliberate weight gain, and all the subjects were very lean, thus having a small pool size. However, when we repeated this experiment in subjects having overweight or obesity, we could again see that a significant increase in linoleic acid in adipose tissue, although the magnitude of change was about half the size, likely reflecting the much larger pool size in these individuals with overweight. So to wrap up, I'd like to give some future directions. When looking at the fatty acid composition and in relation to disease outcomes, typically we look at fatty acids individually. However, there are a handful of studies using, for example, principal component analysis to find these fatty acid patterns, combinations of fatty acids. And one example is from the EpiHealth Interact study, where they did find a pattern characterized by high levels of linoleic acid and also high levels of these very long chain saturated fatty acids and odd chain fatty acids, and lower proportions of MUFAs and other saturated fatty acids. And this pattern of fatty acid was strongly inversely associated with risk of type 2 diabetes. And they also tried to find the dietary determinants for this pattern, and they could see that this pattern was positively associated with the intake of unsaturated fat and fiber, fish, nuts, vegetable oils, and inversely associated with soft drinks and processed meats and alcoholic drinks. However, I think we should spend some more effort in using these fatty acid patterns also in relation to dietary intake, and not only in relation to disease outcome, which has primarily been done so far. Perhaps also in combination with the omics techniques. Another area where we need more understanding is for the minor fatty acids. For example, these very long chain saturated fatty acids, but also for the odd chain fatty acids. In observational studies, these typically cluster together. This is another example from the EpiHealth Interact study, where we can see increased risk of disease for the even chain saturated fatty acid, but the protective effects for both the odd chain fatty acids and the very long chain saturated fatty acids. So they typically cluster together, and in this study, they were also related in a similar way to self-reported dietary intake. Both the odd chain fatty acids and the very long chain saturated fatty acids showed positive associations with self-reported intake of fruits and vegetables, nuts and seeds, and dairy products, and inverse associations to red and processed meats and alcohol, so which is in stark contrast to these common even chain fatty acids. So to conclude, we don't have a biomarker of total fat intake, but we do have good and useful biomarkers of fat composition. And the prime example here is, of course, the PUFAs. And here we typically see a dose-response relationship, and this is seen across circulating lipid fractions. We also know that circulating fatty acid biomarkers typically reflect recent intake rather than long-term intake. We know that if you alter the dietary fat composition, this will rapidly be reflected across multiple lipid fractions, including phospholipids and red blood cells. So it's important to remember that we rely on this assumption that subjects has not made any drastic short-term changes. And I think we should spend more time on trying to look at fatty acid patterns in relation to dietary intake. Thank you for your attention.
So thank you so much for a very interesting talk. I don't see any new questions here, but well, I have one at least. What about total fat? Do you think that we will have a biomarker for total fat, or will that be impossible in the future?
Yes, good question. I hope so, but I think it's not very recent. Very soon we will have that, and I think it's quite difficult. So I don't have very high hopes in the near future, but and I don't think that fatty acid composition, as typically measured now, may be useful for that. But perhaps other biomarkers will. But we'll see.
Yeah. So you don't think you can combine what you have now and get a measure of total fat? No.
Not in a very reliable way. No.
No. Okay. And you also said that you have the PUFA biomarkers are better than, for example, for saturated fats. So what are the correlation coefficients for polyunsaturated fats about?
It depends quite a lot to which fraction you look in, and also, I mean, the specific study, but they can be quite high, but they are not, I mean, they are not super high, but they are still reasonable, I would say, like medium strengths, but it's difficult to give any particular number here because that's where I.
But do you know of any studies where they have combined the biomarkers of fatty acids with reported intake when you look to disease outcome? Has that been done yet, or?
Yes, I think that has been done. I don't have any specific example on top of my head. No.
Thank you so much. Very nice presentation. And yeah, both speakers, thank you so much. And now we will have a break. And we'll continue at 2:20.
I hope you had time to stretch your legs, maybe had some yoga, or maybe have time to take a coffee. I will continue being a moderator for the next session. So I will start introducing Günther Kühnle, professor at Reading University from UK. So please, Günther, the floor is yours.
Thank you very much. I think you can hear me now.
We can hear you.
You can hear me and you can see my presentation.
No, we can't.
Can you see my presentation?
No.
It did work the last time. I'm so sorry. It did work before. It tells me I can't share the screen.
Should we continue with the next presentation, maybe? You can. Now it works.
Now it works. Okay, thank you. For some reason, it stopped me from sharing it. So you can see it now.
Yes.
Great. So thank you first for your very kind invitation to this talk. It's one of the first conference talks I give in some time, and I'm talking about a type of biomarkers which are, from my point of view, extremely exciting, but which are not really used this often in nutrition, unfortunately. And I hope it will be taken up a bit more. And these are stable isotope ratios as biomarkers of dietary intakes. And this is just a picture of the Reading, normally I show a picture of our food science building, but our botanical garden looks much nicer at the moment. So what I'm going to talk about, well, a brief background about stable isotope ratios, what they are, and then I will focus really mainly on how they can be used and what has been used and what are really future applications of these biomarkers. Because as I said, they're not very commonly used in nutrition. They're very, very popular in archaeology, and they're very, very popular in sort of ecological sciences, but in nutrition, they're sort of seem to have sort of a kind of a maverick place that not many people use them. Stable isotopes, very, very briefly, of course, many of the atoms we are made of have different isotopes. So, and what we are interested in here are the stable isotopes. So not those which are radioactive and decay over time, which is a common confusion I experience when talking about this. Most people, when you talk about carbon isotopes, think of carbon-14 and the decay and timeline, whereas we're interested in carbon-12 and carbon-13 or nitrogen-15, nitrogen-14, oxygen-18, 16, 17. So this stable versions of the isotopes. And the reason we are interested in those is that these isotopes can give us a huge amount of information at a compound level. So before we heard about how we can find out what kind of fatty acids people consume, with the stable isotopes, we can go one step further. We can say, where do these fatty acids actually coming from? And that makes this so exciting. So the two main sources or the two main applications of stable isotopes are really to look at different types of plant foods and different types of food where one is in the food chain. The reason we get this differentiation is really that, of course, these different isotopes act very, very similar. Chemically, there's virtually no difference. But if you, of course, do large numbers of reactions, as we have in physiology, then suddenly we find a differentiation. And that's, of course, because the heavier isotopes are slightly heavier, bonds that are formed are a little bit more stable, reactions take place a little bit more slowly, and over time, this causes a separation. And one very nice example for this is the difference between C4 and C3 plants. So those who are not botanists might have to go back a little bit to what I remember about different types of photosynthesis. But C4 plants, essentially plants which have adapted to an environment where it's more difficult to fixate carbon dioxide and which have an additional step for carbon fixation. Now, the normal plants, if you want to call them, have rubisco, ribulose bisphosphate carboxylase oxygenase, which fixates carbon dioxide. And this enzyme slightly discriminates against carbon-13. So these discriminations are very, very small. It's not massive, but they slightly discriminate. And if you look at the graph here, you can see the carbon-13, the delta carbon-13 value, which is a standardized value to show the difference between carbon-13 and carbon-12, is much, much lower in C3 plants like wheat or rice or beans than in C4 plants. And that is very nice for us because C4 plants in our food chain are essentially corn, sugarcane, and sorghum. And especially in non-European applications, corn and sugarcane are quite important because sugarcane is the main source of sugar, and corn is in many parts the main source to feed, for example, animals, but also source for high fructose corn syrup. And you can see in this graph that how nicely these two separate. And of course, if you eat a plant, you will take up their carbons and make human from these carbons. And this is a little bit more detailed. I don't want to go into the great detail of the biochemistry, but it's very fascinating. I'm a biochemist. I find this absolutely fascinating. The other interesting aspect of it is the isotopic fractionation along the trophic level. So the higher you are up in the food chain, the more nitrogen-15 you keep because N14 is excreted, the N15 you retain, and the higher you climb up the food chain, the more N15 you have. And that is very interesting to distinguish between different types of dietary pattern. So if you consume a solely plant-based diet, you're from that point of view very low in the trophic level because it's essentially the plant-based diet in you. If you consume a fish-based diet, you have of course much higher trophic level because fish tend to have a fairly complex system and they sort of start from phytoplankton and going up. And that is very nicely because if you take a hair sample or nail sample of someone, you can very easily distinguish someone who has a high fish diet, and you can very, very easily identify someone who is on a plant-based diet. So that works really, really well, these isotopic fractionation. And this is just really a summary. You can measure a huge amount of things from stable isotopes. You can measure migration patterns where people come because the soil has differences in stable isotopes. You have differences between the equator and the poles where you have differences of deuterium and oxygen stable isotope ratio. So you have a huge amount of information there. But for nutrition, obviously the most interesting are carbon, nitrogen, and possibly oxygen, because that would give us information on the diet. And this first very simple approach is quite easy to apply. You can measure this in bulk. You can measure hair or nail, or you can measure whole blood sample, red blood cells, or even just blood or the albumin in blood. And the further you go up in the carbon-13, the more you see people have consumed C4 plants, corn, sugarcane, and the higher you go in delta N15, the higher the trophic level is. So this gives you already an idea that if someone is very high in both, it's very likely that their diet is probably very corn-based and probably a lot of fish. Incidentally, you can, for example, identify very easily someone who's coming from the US, because in the US, a lot of food is based on corn. So animals are fed corn as well. So you have a fairly, fairly high carbon-13 compared to a European population. But you can see you can use this already to sort of broadly class people into different classes. But how can we actually use this? Because yes, fine, we can distinguish between people who eat meat and people who don't eat meat, but that's not really as exciting as this. So we want to go a bit closer. Can we quantify this? Can we sort of identify how much people are eating? And we have run a few years ago a fairly well-controlled diet intervention study where we fed people a high meat diet, high fish diet, and a combination of these diets. And you can see here these back plots for this. And you can see very easily in urine, we can distinguish between fish diet, between a meat diet and vegetarian diet. It gets a little bit more difficult in feces. Let's ignore this. But what you also can see, this was an 8-day diet. In urine was very, very easy to distinguish between, because within these 8 days, you get a lot of metabolism taking place. So a lot of the food that people consumed will be excreted. In blood, interestingly, we didn't find any difference whatsoever. But that's not really surprising. This is whole blood. So the main bulk of carbon and nitrogen in there are in the red blood cells, and they don't change within 8 days. So from the previous talk, so that the question about long-term and time frames, this is quite important, that by picking the right type of source, we can sort of look at very, very different types of time frames to look in. If you look at blood, it's 60 days. If you look at, so red blood cells, it's 60 days. If you look at the albumin, it's sort of 10, 15 days. If you look at nails, we can sort of go a long time. If you use hair, you can have a very, very long-term, long time overview of what people consume if they do have hair. And this is from actual applications. So this is in a study in Oxford where they compared, again, people who are omnivore and over lacto-vegetarian and vegan. And here you see one limitation of this approach. If someone doesn't eat meat but consumes a lot of animal products such as dairy or eggs, there's not really a difference. So as I said, the difference is mainly between a mainly plant-based diet and not a plant-based diet. You can see fish, because fish is generally much, much higher, but sort of the normal middle of the road type of diet, whether they're just eating meat or vegetarians with fish and eggs, it's more difficult to distinguish. And if you go for habitual diet, it gets even worse. And this is from a study where we had people on their normal habitual diet, and we estimated sort of how much plant food they consume. So this is the proportion of plant food. So it's 100% to about 60%. And you can see, well, the problem we have sort of pies like a fish pie or stargazy pie, which is a British specialty. You can see the differences in a plant food are not really this huge. And this is sort of one of the limitations of this method. If you just go for bulk stable isotope ratios, yes, we can sort of distinguish the extremes. Identifying small differences is much, much more difficult. And that's, of course, something we're often interested in. So it's one of the key limitations we have here. And if you look at longitudinal state changes, so this is one person where we just track this on. So you can see this is a little bit of messy graph. So it's just really sort of to show the differences. You can see here the carbon-13 ratio for urines on the top, and then for the diet. You can see sort of the urine follows the diet a little bit. But what I want to show here, sort of the changes are fairly slow. So if you want to look at major changes, it gets difficult. So bulk stable isotope ratios are very interesting if you want to look at extremes. But if you want to look at sort of more the small changes we're interested in nutrition often, they might be a little bit too crude. And then there's, of course, another big problem, and that is stable isotope ratios are not just affected by diet, but also by metabolism. And this is a nice study by of pregnant women looking at a stable type isotope ratio in hair. And they were collected at birth, and then you sort of back calculated when was the likely conception. And if you ignore this outlier here, you can see that, well, no, you can see that in all women, there's a sort of a drop in the nitrogen 15 value. And of course, if you go from the very, very crude approach and say that if you have you have to feed essentially the embryo or fetus, and that takes away, well, that acts essentially like a type of trophic change. So it takes away some of the nitrogen 15. So it affects your stable isotope ratio as well. And similar effects you get, for example, when people are starving or when people are ill. So when people require intensive treatment, also the stable isotopes are affected. So as I said, the bulk measure is interesting and useful, but is so sensitive to many other things that it might not be extremely useful in nutrition. And that's why it's much more important and interesting to look at individual compounds. So we have the same thing in starvation. So if you starve, your nitrogen 15 values, again, also changes. And as I said about specimen, and depending what specimen you use, you can have long-term and short-term. So single compounds are really, really interesting, because if you want to look at single compounds, that's where the stable isotope ratios really can show their performance. And this is sort of the first study I've seen where this was done in a larger scale. This is bulk, and this is using stable isotope ratios as a surrogate marker for EPA and DHA. But that's really just sort of a fairly, it uses nitrogen 15 as a surrogate marker of fish intake, and fish intake is linked to EPA and DHA in that population. So it works, and you can use this if you know you have a population, you have a huge range of fish intake, but most people would not really consume this amount of fish. This is in native Alaskan people. But if you go, we can go one step further. We can look at individual compounds. And that's, as I said, that's what is great. So this is sugar intake. If you use sugar in the US, it's mainly based on high fructose corn syrup. So people with a high sugar intake, you would expect sort of have a higher bulk carbon-13. But where is it from? Is it from sugar, or is it just simply beef? As I said, in the US, a lot of meat is produced based on corn. So you would expect them to have a high carbon-13 stable isotope ratio, because simply it's based on corn. So we could get one step further, and there it starts to become really fascinating, because now we get into biochemistry. And this is the Cardiometabolic cycle, and it took a long time to figure out how this cycle is called. And essentially what it is sort of the shuttle between muscles and the liver. The muscle exports alanine, the liver does gluconeogenesis and export glucose. So if you consume large amount of sugar, that can be exported to the muscle via glucose and then goes back via alanine. So the carbon skeleton remains the same. And this carbon skeleton is based essentially on the carbohydrate you eat. It can also be based on proteins, but sort of the carbohydrate you consume is quite a large amount. And if you look at the different amino acids here, you can see the non-essential amino acids, that's what your body produces. They're probably, a lot of them are made from the carbons you consume, those who can. The essential amino acids, they must come from proteins. So you can start to distinguish between different amino acids. And if you know that the alanine in blood, and there's a lot of great data on this, the alanine in blood is based essentially on the carbohydrates you consume. You can say if you consume a lot of free sugars, and you know these free sugars mainly come from corn derived or from sugarcane, if alanine carbon-13 is elevated, you know people have consumed a large amount of these kind of sugar-sweetened beverages, for example. And that's where these biomarkers become really exciting. And this is a correlation which you can see sort of red blood cell and hair, alanine carbon-13 stable isotope and sugar-sweetened beverages intake. And you can see a very, very good correlation. And now, since we know that it's very difficult to measure sugar intake, and sugar-sweetened beverages intake. This is really exactly what we can do. So we can even use hair samples to identify how much sugar-sweetened beverages people would have consumed. And this group has done, well, this is a larger collaboration, but the main group is in Alaska. They have done quite a lot of analysis of different amino acids. And you can see that the non-essential amino acids are sort of all very strict. Carbon-13 ratio, non-essential amino acids are strongly associated with sugar intake. So if you measure stable isotope ratio in various amino acids, they give us a very good idea of the amount of sugar-sweetened beverages people consume, but also the amount of meat people consume. And that's sort of where the strength of these markers is. The application epidemiology so far, and this is just sort of my last slide, is a little bit more difficult. We have done this in EPIC, in a subset of EPIC, and we've tried to understand this in bulk stable isotope ratios. And that has sort of, I think that was really, really exciting when we started. And then we analyzed the data, and then there was sort of, so here we have the data, we know what it should mean, but it was very, very difficult to identify because suddenly we had associations with smoking status and other factors, which we have not really thought of what it means. And I think what this shows is that we need more detail, so we need compound-specific stable isotope ratios to really interpret results and go further than just say, well, we have terrestrial animal eaters, fish consumers, which are simply because the differences are not big enough. So in summary, really, these stable isotope ratios are really, really complex markers. Yes, they provide complex information because they not just tell you, for most biomarkers, so if you have a molecule and you trace this molecule back to something, no, you go to the atoms of the molecule and trace back where these atoms are actually coming from. And because they're so affected by metabolism, it's not just that, it's also what fate did these atoms have throughout their life. And that makes it difficult to interpret. But of course, they provide you really, really detailed information, such as carbohydrates. Where do the carbohydrates come from? Sugarcane, sugar beet. You can go further to see proteinogenesis or lipogenesis, different metabolic processes. You can look at anabolism or catabolism. It's very interesting. So, for example, if you look at people who are starving or who have dietary changes. So there's quite a wide range of looking into different aspects of metabolism. And that makes these stable isotope ratios so great. And unfortunately, really, they're completely underused. So we don't know much about how we can really apply them here in nutritional epidemiology. We can, for example, distinguish between different sources of nitrate in urine, so whether it's endogenously formed or exogenously formed. But again, this is sort of things which have been used in other fields in nutrition. They remain fairly underused. And that is the end of my talk. Thank you very much.
Thank you so much. It's interesting that you say that it's been used in other areas, not in nutrition. Is it expensive to measure or?
Well, it's not really expensive. It's sort of, the thing is most archaeology departments or ecology departments have instruments to use this. Some of it is very laborsome. Some of it requires a lot of preparation. But in the end, compound specific, if you have the instrumentation, it's fine to use. It's, I think, having the instrumentation and using it. And a lot of it is not automated. So when we measure large amounts of urine, it means that someone has to really prepare them and analyze them. But really expensive, they aren't. I think it's mainly underused because there's the usual problem we have. We don't talk to people from other disciplines. And then you meet an archaeologist and say, oh, we try to find out what people are eating. Oh, really, you do? We do the same thing. And it's sort of this thing. And I think there's a big opportunity.
Yeah, very good to hear. We 2 minutes over time, and there's no question in the Q&A. So I really want to thank you for that interesting.
Thank you.
And then we continue with Stina Ramne, PhD student in my group, who will present. Stina?
Yes, thank you. I will try to share my screen, and I hope you see the. Wait, you see this, right? And now you see the full screen?
Yes.
Yes, good. Okay, yes, hi, my name is Stina, and I'm a PhD student in Nutritional Epidemiology here at Lund University, and today I will talk about urinary sugars as biomarker of sugar intake. And that is, first of all, because in Europe, we get our sugar from sugar beet and not from sugar cane or corn, which we could measure with the stable isotope ratio that Gunther just mentioned. But here in Europe, we have sugar beets, and it's not possible to use that biomarker. Instead, we can measure urinary sugars. And the other main reason why I will talk about this is, well, it all starts with my main ultimate research question, and that is, does high intake of added sugar increase cardiometabolic risk? And this is a question that most people might think is obvious. Yes, but once you actually look at the epidemiological studies that has been done, you are no longer so convinced. So if we look at all the studies that have looked at the association between sugar intake and type 2 diabetes or cardiovascular disease, most studies have found no association, here in gray. And there's a few studies that have seen harmful association in red, but there are also just as many that have seen protective associations in green. So overall, this is very shaky. And we have some more evidence from randomized control trials, but they are also small and short and only looks at surrogate markers of disease. So yeah, we have some more there, but it's still shaky. And these epidemiological results make you, of course, wonder, is inaccurate dietary assessment contributing to the lack of evidence? And yes, I think that likely is a contributing factor among many. And that makes you wonder, can we measure sugar intake objectively? And yes, we can that too. By measuring 24-hour urinary sucrose and fructose. And this is a biomarker that has been researched for 15 plus years now. And I'm going to start with explaining how it works. Because sucrose, why is it even in the urine? According to all nutrition textbooks, sucrose is hydrolyzed into glucose and fructose before absorption. But actually very, very, very small amounts are absorbed and reaches circulation. And fructose is happily absorbed just as it should be, but it's quickly metabolized in the liver, but not all, some reach the circulation. So now we have small amounts of sucrose and fructose in the circulation, and then we must remember that insulin only regulates circulating levels of glucose, not sucrose and fructose. And therefore, it is believed that once sucrose and fructose are in the circulation, they are excreted in the urine quite untouched, so to say. And since these amounts that are excreted they are very, very small, only about 0.05% of what has been ingested, but those amounts are proportional to the intake, and that's why we can use this as a biomarker of sugar intake. So in super duper mega controlled validation studies, they have seen that the correlation coefficients between sugar intake and the urinary sucrose and fructose biomarker from 24-hour urine samples are very high, over 0.8. And once you instead compare the same biomarker to self-reported intake, the correlation coefficients are much lower, from 0.2 up to 0.5, dependent on the dietary assessment method, but also seems like there might be a dependent on sex. But this reduction from 0.8 to 0.2345, that's due to the data being self-reported here, because the biomarker is measured in the same way. So that's the amount of error that is introduced when we have self-reported data. So when I think of all the literature that we have on this biomarker, two major questions arises that I would like answers to, or actually more questions, but these are the main ones and the ones I will talk about. So first of all, must it be 24-hour urine samples? Because to collect every drop of urine for 24 hours, that's very difficult and burdensome. And especially if you would like an entire cohort of 30,000 individuals to do it, then it would be easier if we just had one urine sample. And the second question is, can this biomarker be used in any population group? Or do we have any limitations there? So if we start with the first question, so there are a few studies that have looked at this biomarker from other urine samples than 24-hour ones. And Tim Inteman, who is on the schedule for this NEON webinar tomorrow, he has found that the morning urine samples, and also this is in children, they seem to work fairly well. There are correlation coefficient between 0.2 to 0.25 with self-reported intake, or I guess they were children, so I guess they had some support. But at least these are in the range as seen with the 24-hour biomarker 2, but in the lower range. So it seems to be working well. And another study that used spot urine samples, and this is a study by Günther, who just performed or presented perhaps before me, he made this study where they found a negative association between self-reported sugar intake, and that's the square ones, and they found a positive association between spot urine sucrose concentrations, and these are the circle ones here. So what you think is that this indicates that this negative association, that's due to dietary misreporting, and the urinary measurement reflects the more true intake and the true association. That's a nice way to interpret this. So last year we published this study where we studied the overnight urine samples in the Malmö offspring study. And overnight urine samples are morning urine samples, but it also includes if you had to wake up anytime during the night and release. Yeah, it was Günther Kühnle who made these LCMS analysis of sucrose and fructose in these urine samples. And yeah, I'll move on. We found correlations with self-reported sugar intake from 4-day food records between 0.21 to 24. So also in the same range as seen before. But we could also see a slight difference between the sexes. And if we look at the same agreement, but in categories of self-reported intake and the urinary biomarker, this is what it looks like. So there's clearly an agreement here, but it looks also very messy, and that reflects what a correlation coefficient of about 0.2-ish plus would mean. But this is what it looks like. So that just visualizes this a bit more. But what we also did was that we looked at both these different measurements of measuring sugar intake with cardiometabolic risk markers, and I've just chosen a few here. And we could see some differences between them and also between the sexes. So for example, both BMI and waist circumference, if we generalize a bit, both the self-reported and the urinary sugars associated positively with BMI and waist circumference in women, while in men, the urinary sugars associated negatively, and we found no association for self-reported sugar intake. And these results are very tricky. I don't really understand them myself, but we considered that since self-reported intake has its limitations, and since this is from overnight urine samples, the urinary biomarker likely also has its own limitations or errors. But importantly is that those errors are completely unrelated. And based on this, we considered that it would be most useful to combine the two into one composite measure to deal with these different errors. So that's what we did. We combined them using their first principal component, and then we could see more confidently, I would say, that the composite measure associated with higher BMI, higher waist circumference, higher systolic blood pressure, and lower HDL in women only. So in conclusion of this study, we found moderate but significant correlations between reported sugar intake and overnight urinary sugars. And we think that the overnight urinary sugars can be used as a complement to self-reported dietary data. But validation studies are needed. We have only compared to self-reported intake. There's no validation study that has ever looked at any overnight or spot urine samples. So that has to be validated. But if we move on to my second question, can this biomarker be used in any population group? And then I think it's mostly interesting to think whether it works in those with metabolic impairment, since we know that glucose excretion in urine is very much elevated if you have diabetes. But on the other hand, insulin does not regulate fructose and sucrose in the circulation, only glucose. So for that reason, it's reasonable to think that having diabetes or prediabetes would not necessarily have an effect on this, on the usefulness of this biomarker. While on the other hand, there has been observations that fructose in urine and circulation is increased in diabetic patients, hypothetically due to endogenous production. And also, we know that intestinal permeability can be increased in patients with diabetes, which could increase the sucrose absorption in the small intestine, which could, so both of these reasons are reasons for why maybe this biomarker is not useful in people with prediabetes or diabetes. So I turned to the preview study in that there were 379 individuals with prediabetes in the Copenhagen branch of this study. In total, there were over 2,000 individuals included across all study sites. But in this study, participants, they started with the 2 months low energy diet in a weight loss phase. And then they were randomized to either a high protein, low glycemic index diet or a moderate protein, moderate glycemic index diet. But what is truly amazing also in this study, I think, is that they at several time points have collected 24-hour urine samples and 40 food records at the same time, allowing for a comparison between 24-hour urinary sucrose and fructose and self-reported sugar intake in pre-diabetic individuals. And also what it might allow us to study is if this agreement between the two variables, if that changes as metabolic health changes. So here are the fasting glucose levels. Just stretch it out to fit the timeline across the dietary intervention. So they lose a lot of weight and of course get better metabolic health after the first 2 months. So yeah, and there's not really a difference here also in success of metabolic health between the different intervention groups. So that's also important to keep in mind. So it might be, allow us to look at that. And I have just received some data, just not even 2 weeks ago, on urinary sucrose only. And actually, I received the urinary fructose data just in my inbox 2 hours ago. And it's Lars Dragstedt who presented first today. He's the one in charge of these analysis of urinary sugars. But I have very, 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 very preliminary results about the urinary sugars that I just wanted to show. So at baseline, there is a correlation between urinary sucrose and self-reported total sugar intake of 0.18. So it's there. It's maybe a bit lower than we have seen before, but it's there. But then at both 6 months and 12 months, there's no longer correlation, and especially not at 6 months. So this sort of contrasts the hypothesis that maybe that the biomarker could be, the performance of the biomarker could be improved if metabolic health is improved. But on the other hand, during 6 months and 12 months here during the intervention, they have a very much reduced intake of food overall and energy and sugar. And they have also just experienced this extreme metabolic change. So I'm not surprised that this is turned on its head. But this is work in progress, and we will learn much more once we have I have looked at the urinary fructose levels too. So if anyone has any input on this, you're very welcome to hit the Q&A or email me afterwards. But this is just an ongoing project. And to summarize my two questions, must it be 24-hour urine samples? Well, it seems that likely it's useful to look at other types of urine samples too, but nothing else is properly validated, so that has to be done. But it seems like that is working. And that's just what Lars said in when he answered the question, that it might be the best way is actually to have maybe many morning samples across several days rather than picking one day to do a proper 24-hour one. And then, can this biomarker be used in any population group? Well, it has been properly validated in children and in people with obesity, so that's good to know. And I'm currently working on the role of this biomarker in pre-diabetic individuals. But I will not be able to validate that either. It's just a comparison against self-reported intake. So that's about as much conclusion I can give at the moment, but hopefully soon there will be some more. So I would like to send some thank yous to my colleagues and collaborators and funders, and I'm happy to answer any questions.
So thank you very much, Stina. I think Emily had problems with her sound, so I'll see if I can find some questions here. So one question for you. What was the methods used in the super-controlled validation studies versus the methods in the self-reported studies?
So I mean, the biomarker was measured in the same way, but in the validation study, there were two of them, and they were 30-day people stuck in getting all meals served exactly, and all urine collected. They sort of lived in this ward where so all foods were given to them. So we had full control, or not we, but they had full control of what everyone ate, and also what everyone left in the toilets. And there was two of them. So there was one dose response study where they had 10 days on one dose of sugar, 10 days on another dose, and 10 days on another dose. And they could see a perfect dose response association. And then the second one was habitual diet, I would say. So when people had already done diet assessments, so they designed studies to fit their usual diet, and then all foods were given to them, and also while being in this ward for 30 days, they collect duplicate portions to get exactly the food they ate. While in the self-reported studies, that was the open study, and the NPAS, it stands for Nutrition Physical Activity Assessment Study. So they had multiple different dietary assessment methods that they used for measuring dietary intake.
Thank you. And thank you for a very nice presentation, and very nice that we got so fresh results.
Yeah, it's in the middle of everything.
Maybe there was one more question now. How do the urinary biomarkers relate to HbA1c as a long-term assessment?
Yeah, and that's that's a perfect thing that I can measure, assess now in this preview study, since we have multiple measurements across the timeline. And I didn't mention that, but I cannot see that HbA1c is associated with the amount of urinary sucrose, but I see actually that this is very preliminary, but that serum insulin is a second predictor of the urinary sucrose. So not only self-reported total sugar intake, but also serum insulin. So, but I have just like super basic analysis so far, so I'm hoping to understand that more.
Okay, thank you so much.
Thank you.
And now we'll have a break again, and we start 3:10.
Okay, welcome back, everyone. I hope you had a nice break. I just want to remind you about the Q&A. It's anonymous, so just post your questions there and I will try to let the speakers know. But now it's my pleasure to introduce Otto Savolainen from Chalmers University of Technology, who will talk to us about Dietary Biomarkers 2.0. So please, Otto, go ahead.
Do you see the presentation now?
Yes.
Yes, great. Okay, but thank you for the introduction and thank you for the possibility to come here today to discuss more about the food intake biomarkers project that we are working with at Chalmers. So my name is Otto Savolainen and I'm working at the Food and Nutrition Science Division at Chalmers in Göteborg, and I'm part of the research group led by Rickard Landberg, where we have a strong focus in both health and diet and also on food intake biomarkers, both when it comes to application of food intake biomarkers and also on how to use and how to develop methods for measurement of food intake biomarkers. I'm also working as the head of the Chalmers mass spectrometry infrastructure, and we collaborate a lot with the mass spec infrastructure in this project, as we have good instrumentation available through the mass spec for these applications. So we already got a very good overview in the food intake biomarkers by the previous presentations, but I will want to highlight a few key points that I see that are important for our application. So as we all know, diet is a major risk factor for disease and mortality, but most importantly, it is a modifiable risk factor, which then provides means for us to intervene into the development of disease or intervene into the disease development trajectories that we have in the population. And we also have a very good set of tools available for measurement of diet in the research setting, for example, in cohort studies. But all of these tools comes with inaccuracies and are subject to many sources of bias, and we are well aware of this. So certainly we have the possibility, or there's a room for development or addition of tools in the toolbox that we can use to measure diet. And the existing tools are not very well applicable to a routine healthcare setting, where we could perhaps want to measure diet at the one point of time when we have the subject at the clinic. So taken together, if we have more tools available, it will add on to our understanding of diet and health. And it will also increase our understanding of what is the habitual diet that we are consuming. And then food intake biomarkers here could provide one means to improve on our measurement of dietary intake. So during the last one or perhaps several decades, we have had a very large focus on the discovery studies when it comes to food intake biomarkers. And we look at the literature, we have literally hundreds of potential biomarkers reported. So we have a quite large pool of molecules that may or may not function as dietary biomarkers. And Metabolomics, development of Metabolomics in part has made it possible. So when we have the option, or Metabolomics has become a standard tool in research labs, it has made it possible to analyze the intervention and cohort studies that we have done and relate the small molecule data from the Metabolomics experiments into the dietary intake. So we have used a lot of intervention studies and cohort studies with available dietary data, and we have went out and done Metabolomics with the existing biofluid samples. And then we have screened for associations between the small molecules we have detected in the Metabolomics experiments and the dietary intake. And then we have used these associations to report potential food intake biomarkers. So we have had very little focus on the validation of the multiple markers we have reported in the literature. I think Laras gave a very good overview into the validation status of some of the biomarkers, but when we look into the literature and we see that there's a huge amount of these molecules available, which we don't even know half-life for. So I think there's a lot of work that we need to do to improve on the validity of these markers. But the fact that we have found significant statistical associations between the markers and the food intake, this is already suggesting that we have a large potential for these molecules to be used as a standard tool in the future. And one part, what is missing from the equation of improving on these markers as a standard tool is proper methods for measurement of these dietary biomarkers in biological samples. So if we can exchange the Metabolomics box that we have used for discovery of these potential food intake biomarkers into a standard assay, which is robust and can provide us quantitative information on these markers, we can then go back, we can first go back into the studies that we have already done, and we can design new studies for both confirming the discovery that we have done in the Metabolomics experiments, but then it will also make it possible to start the validation of these markers as a food intake biomarkers. And then also in the other direction, so if we have a good readout of the molecule and we know that these markers can work as a concentration markers, then we can take this data that is provided by the method and turn the data into an actual habitual diet and look into how we can reflect the content of the diet with these methods. So both, of course, as a detection method for detecting if the subject has been eating a food item or not, but then also relating into how large proportion in the diet we are consuming these food items. So this brings us to the project that we are working at Chalmers at the moment, where we are trying to do exactly this. So we are taking the large body of biomarkers that have been reported in the literature, and we are aiming at turning these biomarkers into a tool that we can use to reflect food intake in the biological by measuring the biological samples that we are collecting from our studies. So the project includes a method development part, and our goal has been to cover over 10 food items or food groups in one method, in one go. And the second part of the project includes, or we have the possibility to conduct a diet intervention study, which we can use both to validate the method we have developed in our lab, but then this will also allow us to start to look into the dose-response relationships of the biomarkers that we have included in our method and that we will include also into the intervention. So this gives us a very good opportunity to both validate the method and then look into the validity of the biomarkers. So starting from the method, so we have been working with development of the method in our lab, and we started the development on a LC-MS platform, not least due to the fact that many of the discovery studies have been done in LC-MS-based Metabolomics platforms, which already is suggesting that we should have good grounds of detection of these molecules with an LC triple quad based method. And also when we went into the literature and we had refined the biomarker lists and collected the information that we want to include in our method, we found out that indeed these can be well detected with the LC triple quad based method. And we are all the time aiming at building a rapid and sensitive method. And we want to do it quantitative, so we see that it's important to have a quantitative readout for the biomarkers in order to translate the method into a standard practice. So what we ended up with is a method that is 6.5 minutes long, including the equilibration time. And we included a total of 31 potential food intake biomarkers into the method. And these 31 biomarkers reflect 21 foods or food groups. So the combination of the relatively quick analysis time of 6.5 minutes that will translate into roughly about analysis of 200 samples per day, and the fact that we can cover well over 10 foods or food groups with that method. So this makes it we see that it makes it a very efficient tool for measurement of food intake biomarker panels. For example, in cohort studies where we have had a lack of tools in the past for these kind of measurements. And at the moment, we are still waiting for the intervention study to further validate the method and look into how well we can quantitatively describe the intake of the foods with this method. And then we also have in this project, we had a great opportunity to design a diet intervention for both validation of the actual method that we developed, but also into start to look into validation of the biomarkers that we had included in the method. And we have planned this intervention and we are waiting for being able to start it. So we have been a bit delayed with the global pandemics, but now we are getting very optimistic that in the fall we get to conduct this intervention and move forward with the validation of the LC-MS assay. So how we have designed this intervention is that we have included 3 different study weeks. And on each of the study weeks, we will provide the subjects with a meal model. So we will provide the subject with ready-made meals. And what is different between these 3 different weeks, I should have mentioned that on these different meals, meal 1, 2, and 3, we are including the same food ingredients on each of the meal. But what is different between these meals and the study weeks is that whereas in week 1, when we are providing the subject with meal 1, we have a very large proportion of food A included in the diet, and then we have a smaller proportion of food B, and then the smallest proportion for food C. And then when we move further down to second week, when we are providing the subject with meal 2, then we have decreased the proportion of food A on the plate if you compare to week 1. And then we have also for food B, we have a smaller dose than on week 1. And then food C is the largest proportion on week 2, whereas in week 1, it was the smallest proportion for food C. And we varied similarly for the week 3. So this allows us to start to look into the quantitative dose-response relationships for these food intake biomarkers. So what is good with this is that we have a fairly accurate picture on the actual intake of the food. And then we have a quantitative readout for the food intake biomarkers with the new LC-MS assay. And this allows us then to look into the quantitative dose-response relationships and start to look into the modeling of the putative biomarkers in humans. And we are also including a single meal study at the beginning of each week, where we are collecting repeated samplings, which will allow us to look into the half-lives of the molecules, as there are many molecules that have no reported half-lives so far. And this will then also feed up into the possibility of modeling the biomarker responses in humans. So with this, with the new assay and the new intervention and the new information that we gain from the food intake biomarkers, we are hoping to start to fill the gap between academic research and the what is required from a method if we want to translate it into a clinical or a commercial setting. So food intake biomarkers has been very much focused on academic research so far, but I think there's a potential also to translate this into applications and to put it into use in the future. And also the fact that the wide implementation of such a method is possible as the mass specs are becoming or have become a very standard tool in the clinical laboratories. So as long as we have robust and good methods available that we can provide and there's a need for the method, then these methods can be implemented widely. So perhaps one day, not too far in the future, we can order a food intake dietary food intake biomarker panel together with our more traditional clinical parameters, and we can use all of these data together in order to look into the disease development trajectories in subjects. And if we have such methods available, this will also allow us to have new means to improve our dietary counseling at healthcare setting. And of course, also provide tools to follow up the development of the diet at the individual level. With this, I would like to acknowledge the whole Chalmers team. So there's some of the key persons working on the project listed here, but there are also a lot of other people at the division that are making up for the good working environment that we have. I would like to thank the Chalmers Mass Spec team for providing good instruments in good conditions for this project, and the collaborators for all the great work that they are doing with us with the food intake biomarkers. And with this, I would like to thank you for the opportunity to be here today.
Thank you very much for this wonderful presentation. So if there are no other questions, I would like to ask you about the validation part. So what is the most challenging part when it comes to validating? Is it the samples or the machines, or what are the challenges in validating these types of biomarkers?
I think because we have it's very many molecules included in the method, and then to validate the marker as a concentration marker, so that we have a quantitative response in the biomarker when we know that we are increasing the dose of the biomarker. So I think this will be challenging as such, and then also when we are transferring into a more high throughput measurement setting with the method, that will of course most likely bring challenges on the instrumental side.
Okay. Thank you very much, and I will look forward to having this kit that we can just test, and we don't have to ask people and they don't have to lie. So thank you very much, Otto, and we will move forward in the program. And the next speaker is Filip Ottosson, who is a postdoc at Lund University. And he will speak about metabolomics and the dietary patterns in Malmö cohorts. Please, Filip, go ahead.
Thank you very much. So I hope that you can hear and see me at the moment.
Yes.
So, and thank you for inviting me to speak here today. So I will speak about our metabolomics measurements in the cohorts in Malmö, and how we have used those measurements in relation to dietary patterns that we have in the same cohorts. So just to give an overview of my view of the human metabolome, the human metabolome is influenced by several factors. So genetics, of course, but also dietary and environmental exposures, And them all in concert, they work to form our metabolome. And then our idea is that we can use the metabolome in order to tell us something about the risk we have of developing different diseases. So in particular, maybe the cardiometabolic diseases, but also it could say something about our lifespan. So what I will speak about here today then is more this connection here, how the diet that we eat, how it influences our metabolite levels, and how those metabolites that are influenced themselves are related to disease. So just to give an overview of what we have been doing here in Malmö, we have measured, we have metabolomics measurements in 3 different population-based cohorts. So it's the Malmö Preventive Projects, which had a baseline in the beginning of the 2000s. And then we have the 2 cohorts where we have dietary data. So it's the Malmö Diet Cancer study with a baseline examination in the beginning of the 90s, and the Malmö Offspring study, which is still ongoing. So we have measured metabolomics in over 8,000 plasma samples using our LC system. And we have used both like a non-targeted approach, like measuring loads of metabolites. But what I will speak about today is more like a semi-targeted approach, where we have a data set where we have all the metabolites that we have correct identifications for using chemical standards. Those are a bit over 100 metabolites in this data set. So imagine that we have a good way to measure healthy eating. So we can get a measurement of healthy eating. Then we see the room to use metabolomics measurements in 2 different ways. So the first one is to find, we can use that healthy eating measurements to find healthy metabolites. And that would work something like this. So healthy diet will give rise to a change in the levels of metabolites in our circulation to increase or decrease, and those metabolites could then potentially have an influence on our disease risk. The second approach would be more to use the metabolomics as some kind of proxy of healthy diet. So you could train a metabolite model using your metabolomics data to describe healthy diet, and you could get like a profile, and that profile could be used in the future maybe to get personalized dietary advice just based on blood sample. So if we move to the first kind of approach, we have used collaboration here with Ulrika Eriksson, and she had in the Malmö Diet and Cancer study, she used a data-driven approach in order to identify food patterns. And she found that the food pattern that described most of the variation in dietary intake was the so-called health-conscious food pattern, as she called it. And that was dominated by high intake of fiber-rich bread, fruit, vegetables, and also fish, and a bit lower intake of low-fiber bread, meat, and sugar-sweet beverages in general. She found that following this pattern was associated with a lower risk for type 2 diabetes, but also coronary events and strokes. So what we wanted to do is to find metabolites that were associated with this health-conscious food pattern. So our very talented PhD student, Ina Smith, did this analysis. So he conducted linear regression models in the Malmö Diet and Cancer study. In the part here, both metabolites measured and dietary data available. So he found that there were 7 metabolites that were quite strongly associated with the health-conscious food pattern. The one in the top is ergothioneine, and then followed by proline, betaine, and methylproline. they are all metabolites that have been shown before to be correlated with higher intakes of either specific vegetables or fruits. And we found similar things here, which was reassuring. So ergothioneine, for example, was correlated with higher intake of vegetables. So our next step then was to see if these 7 metabolites themselves were related to increased or decreased risk of future disease. So we found that ergothioneine was, so being in the lowest quartile of ergothioneine, was associated with a decreased, with an increased risk of coronary artery disease, also overall mortality and cardiovascular mortality. And we also saw that conducting multiple variable adjusted Cox regressions, these associations remained. So ergothioneine is a sulfur-containing amino acid, and it has been proposed to have antioxidant effects before. It has a long half-life, and it's known to be present in mushrooms and vegetables. So our hypothesis here is that this metabolite has some kind of health benefits. Of course, we cannot prove that using an observational study, but we hope to perform intervention studies in the future where we can follow this through for a bit. So if we move on to the second approach, to use the metabolomics as more like as a proxy of healthy diet. So we used the health conscious food pattern that Ulrika calculated in the Malmö study. So this was done in the same way as in the Malmö dietary cancer study. So it's data driven, and this pattern of dietary intake was hello, can you see me? I think something happened here.
We can see you.
You can see me. Okay, so it's something came up here. So you were able to follow me until now, I hope.
Yes.
Okay, good. So I'll continue here. Something happened with my screen. Sorry about that. No, so this health conscious food pattern looks very similar in the Malmö study. So basically, fruits and berries, vegetables, and legumes, and lower intakes of sugar sweetened beverages and meat. So here this time we wanted to use the metabolite measurements in a partially squares regression model, PLS, in order to predict the health conscious food pattern. So doing that, we saw that on the y-axis, we see the metabolite signature that we get. So this is the first component from the PLS. And on the x-axis, we get this is the health conscious food pattern. So we see that we have a quite decent correlation between the metabolite signature and the health conscious food pattern. And if we look at the loading plot, we see that many of the metabolites that pop up in the MDC in the previous study also are relevant here. So ergothioneine again, and proline beta. We see also that beta carotene contributes a lot. And on the other side here, we see negative contributions from, for example, the branched chain amino acids that's been related to obesity and diabetes in many studies. And if we look just quickly at the correlation, we see with specific food groups, we see that the metabolite signature and the diet score here, which is the health conscious food pattern, they kind of correlate with the same direction and about the same strength, which is of course expected. So in our next step, we wanted to use the model that we trained in the mom offspring study to see if we could use it to predict the health conscious food pattern in the Malmö diet and cancer study. And when we did that, we saw that we got a quite a bit lower correlation coefficient. So it's 0.2 instead of 0.56. But this also was quite expected. One have to bear in mind that the baseline examinations here were done more than 20 years apart, and that the pattern itself is slightly different, although it's very similar between the two different cohorts. But it still seemed that it proves that our metabolite profile can be used in more than the cohort we trained it, which is good. Also, when we look at cardiovascular risk factors and diabetes risk factors, we see the metabolite signature here in the mom offspring study in the top were correlated with increased levels of HDL, that they were higher in females compared to males, but they were inversely correlated with BMI and triglycerides. We see the same pattern of this correlation if you look at the health conscious food pattern, and we see basically the same correlations and directions with the risk factors in the Malmö diet and cancer study. So what we wanted to do next was to see if the health conscious food pattern itself was associated with disease risk. And we saw that it was. So actually here being in the lowest quartile, or quintile actually, was associated with an increased risk of type 2 diabetes over a follow-up here of over 20 years, but also with major adverse coronary events. And finally, we wanted to use to prove that this kind of method can be used in a cohort where you don't have any dietary data available. So we took the method that the model that we trained in our marketing study and applied it to the Malmö Preventive project, where we don't have any dietary data available, but we wanted to show that this health conscious food pattern itself was related to disease. In doing that, we saw that in the first two models here, the first is unadjusted, we saw that health conscious food pattern was associated with a low risk of diabetes. Also when in model 2, when we adjusted for age, sex, smoking, physical activity. We saw the same thing for also in the Malmö diet and cancer study, and the same thing for major adverse coronary events in both the cohorts. So to conclude, using the first approach, we found early thiamine as a candidate biomarker for health conscious eating that was associated with a low risk of coronary disease and cardiovascular mortality. And we also showed that metabolomics, profiling using metabolomics can use as a proxy measurement, and that it's associated with lower risk of future diabetes and coronary artery disease. So thank you very much for listening, and if you have any questions, I'll answer them.
Thank you very much, Philip. And I'm sorry, there was questions here in the Q&A that the images disappeared, but for me it didn't. Therefore, I did not interrupt anyone. I'm sorry about that. And the sound has also been a little bit shaky, but I think you heard most of it. It's fine. I could hear it at least. We have one question here in the Q&A. What do you think are the most important things to notice when you develop a metabolism signature?
Yeah, so I think if we want to apply it like to measure healthy diet in our case, I think our method could be improved quite a lot, because we don't have targeted we haven't targeted specifically metabolites that are related to diet. So we have like more a broader panel, and some of them are related to diet and some are not. So I think one thing that could be done, of course, is to use more like an approach that Otto described just now, to have more a targeted approach where you measure only metabolites that are, are dietary related. And then I think they could say quite a lot about, they could be much more useful, I think, for this purpose.
Yes, there is another question. I think it relates a bit, but so metabolomic profiling can be used instead of standard diet assessment?
I think in the future, for sure. I mean, right now, there's a lot of validation to be done, of course. And as I said, again, I mean, the method that we use, this is more intended as like a proof of concept than me saying that this is the method that will be used, I think. But if we improve and we more like specify our MS measurements in order to measure relevant metabolites, I think it could be used. And hopefully, if we can do that in just using one method, and if that is a quick method, then it could be very powerful, I think.
Nice. Is project 2 published? So let's see what project 2 would be.
So it's not published. So it's ongoing, and so we are happy for any kind of input that we could get to improve our analysis. We're in the middle of that. So just the first study is published.
Yes, and then there is a last question. So ergothioneine showed strongest correlation to vegetable intake, but somewhat lower to the examined subgroups. Do you think that the most vegetables contribute to the association with the vegetable intake?
I'm not sure if I understand the question. Is it also in the...
No, it's a question that we've got beforehand. So I guess that if you think that all vegetables contribute to the vegetable association, or if there are subgroups that are more or less.
Okay, so
I think that's the question.
For ergothioneine in particular.
Exactly.
So it's known that it's very it's present in very high amounts in mushroom, and that's really the main food that you get it from. And so I think that's why it's correlated with vegetables in general. Is probably that people eat a lot of vegetables, probably eat mushrooms as well. So I think that's the main part. And then I think it's also shown that it is present in other vegetables too, but in much lower amounts. But I think it's mainly reflecting the mushroom intake. I would that was my guess, which I don't have any proof of at all.
Okay, great. There is no more. There's also ergothioneine wine association. That's very strong.
Yes, that's true. So it's
the grapes maybe.
Sorry.
Grapes then, or?
Yeah, either grapes or it's if it's come from like fungi somehow. I don't know. It could also be. We have discussed that before, and we don't really know. We found that it's very strongly correlated with alcohol intake in general. We found that it's really driven by the wine. So it's something that we have to. It would be a nice intervention study if nothing else to do. So we hope to plan that shortly.
I'll sign up.
Yeah.
All right. Thank you very much, Philip. And that was the last talk of the day. And I will now hand over the word to Ulrika again.
Okay, thank you so much, all presenters, and also all the audience for listening. I think it was a very nice meeting today, and I really think that we have understood that there is a lot happening in this field, so that's very promising. And I look forward to hearing more tomorrow. So thank you very much, and we'll continue at 9 tomorrow morning. So hope to see you then.